Good afternoon. My name is Jose Contreras Vidal. I'm at the University of Houston Center Director for IUCRC Brain and a co-PI for the Music and the Machine Research Network. And I'm very, very glad to talk to you today about mobile brain body imaging technology and how we can use that to deliver and inform music-based interventions. Please use the chat if you have any question at any given time, and we'll be happy to answer. This is a two-part webinar, so I'm gonna talk first, and then we're gonna move to my lab and have some remote hands-on demonstrations of the technology. So with that, let's start. Talk about that. Just to acknowledge the support from the Brain Center, NIH, and NSF, Mission Connect, the IEEE. And these are some of the learning objectives that we would like to meet today. First, understand the challenges and opportunities for emerging wearable technologies to deliver and inform music-based interventions, particularly about mechanisms by which music-based interventions act on the brain and behavior. Also understand the importance of technological convergence and team science in the development and use of devices for music-based interventions. And something that is really important as we enter this area of technology to inform MBIs is to understand the challenges in measuring and quantifying extracranial electroencephalography or EEG for using diagnostics and also as endpoints for measuring the efficacy of the interventions. Some of the research questions that probably we share is how does music-based interventions engage the brain and the mind and promote brain health and wellness? I believe that to answer this question, we need to advance understanding of individuality and social cognition in ecological settings. We need to enable the design of medical devices for delivery of music-based interventions. And we also need to talk about the human experience. How are these technologies changing the delivery of these interventions and the whole experience for the participants and the therapists or professionals? This is important as we would like to use these devices to promote brain health, change behaviors, and ultimately promote human flourishing. These are some of the major challenges that we anticipate in using technology to inform MBIs. Of course, the most important is efficacy. The efficacy of music-based intervention depends on scientific understanding of neural variability and individuality. Consider that in the same way that individual variation in gene sequences makes certain drugs more or less effective for some people, giving rise to the need for pharmacogenomics, individual variation in brain responses to musical stimuli will also affect their efficacy. That is, music that has a therapeutic benefit for one individual may not necessarily translate to another. And I believe that to address this challenge, we need to acquire real-world data in ecological settings due the MBIs to quantify brain responses on understanding mechanisms. In other words, we really need to partner with engineers and technologies and others to address this challenge. There are some opportunities here. Medical devices that interface with the nervous system for monitoring, diagnostic, therapeutic, or restorative purposes are a major focus of innovation and investment in the neurotech and medtech industry. This is going to be particularly important for us working in the field of music-based interventions, as research funding might be constrained, but industry can help us to overcome that challenge. Now, for reasons of safety and patient acceptance, it is desirable for these interfaces to be as non-invasive as possible. Moreover, there is a renewed effort to quantify the extracranial EEG using machine learning, deep learning, or artificial intelligence. Another important and timely point here is that different technologies have been developing and growing at an accelerated pace, and they're converging. And this convergence of technologies is likely to accelerate the translation and applications of devices for movement-based and music-based interventions. So I think this is a very timely effort. Now, I'd like to propose a suggested approach to address these challenges. So in terms of music-based interventions, at the top of this pyramid, so we are capturing the context, and that context may also involve the movement that is that accompanies, music listening or active music production. On the other hand, we have the wearable EEG, Internet of Things that allows for communication, interconnectivity between different devices and measuring technologies, and other wearables that we may already carry in our body, as well as the impetus in the development of brain-computer interfaces that can serve as the mechanism for delivery of these interventions. On the other hand, we have the advances, accelerated advances of particularly AI, artificial intelligence. So when we put together these ingredients, music, technology, and AI, I think that is an opportunity for us to understand neural individuality and variation. Now, if we look at using technology to learn how music engages the brain, we can get information about how music changes brain activity, neuromodulation, how the different parts of the brain communicate with each other directly or indirectly through functional mapping. And also understand the context, the mood, the emotions, and all the factors, the stress, all the factors that may change the brain activity as these interventions are being delivered. If we look at the intersection of AI and BCI EEG, we had an opportunity for the development of digital twins, computational models of the music-based interventions and their effect on the brain, the design of or identification of biomarkers or diagnostics based on EEG and other physiological signals. And ultimately, the development of closed-loop medical devices for music-based interventions that empower the user to take control of their own health and well-being. If we combine this information that we gain through technology about the effect of music in the brain and develop models and computational tools to analyze those data sets, I believe we will be in a great position to personalize these interventions rather than delivering them ad hoc. So the other aspect is that conducting these the recordings of music-based intervention in ecological setting is going to lead to unique data sets in terms of real-world data on one hand, but also being able to capture the importance of social engagement during the delivery of these music interventions across the lifespan, and also leading to a better understanding of how we can promote human flourishing in general. So this is a suggested approach, and of course, as we develop this network and we connect with each other, we can refine this approach. Now, I thought that we are in a timely point where technological convergence may lead to accelerate development in terms of using the technology in application for music-based interventions. And one of the oldest definition of a brain-computer interface by Jonathan Wolpe in 1991 is that provides an additional communication channel of the brain with the world. And that means these technologies can be used to talk to virtual, digital, or physical machines, including, the delivery of music. When you add to the BCI's connectivity, that's the Internet of Things, with the AI and the machine learning, deep learning approaches, then we can think about thought control of digital and physical environments. Of course, that includes music, and also promoting cortical plasticity, which have been shown already as an important contributor for neurorehabilitation and speech communication in BCI. So I believe that this confluence of technologies are going to be very important as we develop devices for delivery of music-based interventions. Now, we're not talking here about EEG, but I'd like you to compare briefly, the signals that we get from scalp electroencephalography with respect to other modalities that have been used to study the auditory system or the effect of music. And at the top, so this is a slice throughout the brain, and at the top, you see an electrode of an EEG system. And of course, is embedded within, the medium, in this case, this hair. We have the scalp, the skull, all the hard bone, the soft bone area. We have the cerebrospinal fluid here, and other layers. Which means that if there is brain activity in cortex, the aggregate activity from a very large number of pyramidal cells in cortex needs to propagate through the medium and cross different layers until it reaches through volume conduction the tip of the electrode. Now, all these layers have specific conductance, or basically characterizing the level of distortion that that particular layer is introduced on the signal. So the signal that eventually reaches the electrode, it gets distorted as it's traveling, right? So that means that the EEG signal we're going to be recording on the surface of the scalp gonna be affected in terms of the quality and the level of the distortion, right? Now, if we are looking at an endovascular sensor here, for example, the stent rod from Synchron, right? Is internal, it's inside the brain, so it's an invasive method, but the signal has to travel less layers, right? So it doesn't need to cross the skull or the skull or the scalp or even the hair, right? It is sitting there, and so the signal path is shorter, and therefore it's less distorted. So the signal recorded here will be a stronger signal than the one recorded by EEG. Now, if we go to a fully implantable microelectrode array like the M1 implant from Neuralink, where we have up to 1,024 wires or electrodes that are penetrating cortex, then there is the signal is recorded on site, right? So it's the highest resolution and highest spatial and temporal quality that you can get from the signal, right? So EEG doesn't provide the same signal quality and other type of electrodes. However, it's non-invasive, and you can have full coverage because you can put these electrodes across all the brain, as compared to other options where you have only specific parts of the network that you can interrogate. So, and there's a lot of information on the biophysics of this signal propagation and equations, and that people have used to be able to localize these signals inside the brain when the recurrences are happening outside the brain. Now, this is an example of a brain-computer interface from my lab. We have a participant who is a bilateral amputee wearing a 64-channel EEG cap. And so we are recording these signals, and he's using a decoder, machine learning model that is interpreting those signals and then sending the output to control the hand directly. So as the participant has vision, so he can see the location of the object, the shape, the size, the orientation of the object, his brain can estimate those signals that are needed to achieve a grasp. And so he's able to control this prosthetic hand by thinking about it, right? So is this technology, which I believe can help you to develop ways to interrogate the brain during a music-based intervention, also to produce a way to deliver those interventions. This is another example where the brain-computer interface is controlling a robotic walking skeleton, and so the BCIs have been developed to extract information about intent for movement, and also other type of signals that relate to stress or emotions, and another type of application. So these are just some examples of to visualize what is the maturity of this technology in terms of being able to read these signals from the outside, and also the level of information you can get from those signals to interact with the environment. Now, another important aspect that I think will be very critical for as we deploy this technology for music-based interventions is that it provides a window to study brain networks and how they may change due to the intervention. And so this is an example of, on the left, we have a participant that is a healthy participant, full of able, and on the right, we have a person with spinal cord injury, right? And they are receiving training on how to use the brain-computer interface to control the walking exoskeleton. What is important here is that this intervention is delivered over a period of a month. So there were 9 sessions over a period of a month, and and these skull maps represent, in terms of the color coding, what's the relevance of different skull areas to the control of the exoskeleton, right? So what we see that for this participant, it takes about 5 sessions, about 2 weeks, for the neural representation that is being used to control the robotic exoskeleton to stabilize, to become stable. And we can see that from session 5 to the end of the experiment, we see scalp areas associated with the neural representation for gait to be engaged and important, relevant for controlling the exoskeleton. And so the brain is changing during the intervention, right? And we can see here on the plot on the left, if we plot session number 1 through 9 versus the overall accuracy in the control, in the prediction of the BCI to control the exoskeleton, we see that the participant is improving with practice, right? So the brain is learning to control better the exoskeleton because the decoder that was used initially at session 1 did not change anymore. Now, if we compare that with a person with spinal cord injury, we can see that something similar happened in the sense that the neural representation for the control of the exoskeleton is changing and requires about 5 sessions to stabilize. However, the scalp areas that this participant is using to control the robot are quite different with respect to the control, meaning that probably the lack of afferent signals, feedback from the legs, and also the fact that the brain is disconnected from the spinal cord due to the spinal cord injury, that the network that is now learning to control this robotic exoskeleton is different. And nevertheless, the participant gets better with session, right? So this, again, an example of both how you can use EEG to track changes in brain networks, and second, using EEG to control a machine for delivering the intervention, in this case, the robotic exoskeleton. So we would love to apply this technology to both the delivery of music-based intervention and also tracking change in those networks that are affected by such interventions. Now, so what are the opportunities and challenges? So I mentioned already that I believe the first challenge is capturing real-world data as we conduct these music-based interventions. So the mobile EEG recordings allow us to study the neural dynamics of music individually or in groups with an exquisite temporal resolution in the order of milliseconds. And also in ecological settings, at a theater, in a music studio, at the clinic, at home, church, in any other place where this music-based intervention may occur, which are simply not possible with other techniques. Here is an example of studies that we have been conducting over the years in unconstrained environments, including jazz improvisation, and we talk a little bit about that, but also in other tasks that involve music, like dance, piano, and we have conducted studies also with children. So so it is possible to run these experiments on the wild, and I would like to talk a bit more about that. So this is an example of the activity that we have conducted. Okay, so let me go through some of the challenges that we need to be aware, right? And so the first one is logistical, I would say, and that includes selecting an appropriate, form factor, considering the usability of the devices, the setup time for donning and doffing the devices, the head type, hair type, and the level of familiarization of the researcher with the device setup. And here in this slide, I'm showing 5 of possibly more than 150 devices that you can buy nowadays. So this is just a small sample of form factors that you can find. And you can see they vary in terms of the number of channels. In this case, we have 4 just in the frontal areas of the head to a full coverage of the head on the right, and intermediate levels, right? And we also have, elastic fabric based caps. We have bands, and we have dry electrode, helmets. Again, these are just 5 examples of the type of EEG devices that you can get nowadays. Some of these contain other sensors. For example, the first 2 contain accelerometers that allow you to track the movement of the head as you listen to music in this case, or maybe you are performing and you're able to extract rhythms from the movement of the head. Now, however, they do not perform the same level, right? So on the right is the data rejection rate in percentage, right? In terms of the reliability of the data as the participants were walking through an exhibit in a museum setting. So we can see that, 2 of these headsets on the right, and also with active electrodes and a cap based on fabric, had the lowest data rejection rates compared to the others. And so you need to consider that, what is the amount of data that you are willing to remove because of the quality of the data or artifacts that you might find. So not all headsets are the same. So that's one of the first aspects, right? And it might be related to the study population. Another type of challenge is identifying and removing physiological and non-physiological artifacts, both online and offline, which might be due to the power line noise, 60 hertz, very common for headsets to be contaminated. The recordings with 60 hertz. Probably with device connectivity, with Bluetooth, you might lose some package of data. Have physiological artifacts due to eye blinks or eye movements, shift in the electrode potential due to humidity, temperature, the gel property that you are using, or the lack of gel if you're using dry electrodes. The motion, as you perform or listen to the music, in an ecological situation, it might be important to track head motion and to be able to detect motion artifacts. And sometimes you might have electrode pops where an electrode get disconnected, and therefore it's not able to measure the activity. You might have also perturbation, touching the sensors, handling the sensors during the recording, that all may affect the quality of the signal. These are some examples of errors that we typically find. At the top left, we have the digitization error. You can see that in this particular segment of data from electrode located in the frontal central left part of the brain. we see some clipping here and some errors in terms of the quantification of the signal that we need to remove. In B, we have wireless data loss. So we have a segment of data that we just lost due to lack of connectivity, Bluetooth or Wi-Fi. So that means that data is not usable. Sometimes we have poor electrode contact, and you might check the quality of the signal at the beginning of the experiment, but nevertheless, due to movement and other factors, the electrode may develop, poor contact, and then this is, some of the signal that you will get during that period. So you're missing the information that you would otherwise get if the electrode was making contact. And this is an example of an electrode pop. Sometimes that happens too, and so you need to recognize that either visually or through automated means to be able to either interpolate that period of signal based on adjacent electrodes, or just remove that segment from the analysis. So this is another headset. This is an experiment we did at the Children's Museum. It wasn't a music intervention, but just to show you, different form factors and different applications that can be adapted to music-based intervention. So this is a Muse headset. It has 4 EEG channels and accelerometer. So you can see the children wearing the cap, the band, as they were playing on the computer Minecraft, a game. And the accelerometer data, which provide head motion, head tracking, can inform about their behavior, right? In this case, there's a head shake of the child, right? If he's moving the head to the left or to the right, then you will see those movements. And then the 4 EEG signals that are recorded, 2 in the front and then on the left and right temporal side, you can see here how the head shake affected the EEG, right? And there are some eye blinks here detected that contaminate the EEG signal. And this is clenching of the jaw, right? Which distorts very badly the EEG signal, right? So at minimum, I believe you need to record EEG and head motion so that you can at least have a reference to look at the effect of motion and the potential effect of development of motion artifacts. Now, because different headsets have different type of sensors or number of electrodes, it has been complicated to develop a common pipeline for pre-processing the data, identifying and removing artifacts. For this particular application, where we had 4 EEG sensor and accelerometer data that provided head motion in the X, Y, and Z axis, we developed a pipeline which is on the right. In this particular headset, we were recording at 220 hertz, and we had to remove some channels that we lost, and that was particularly because these headsets were not designed for children, and so they kept moving in some of the participants. So we had to remove those channels. We decided to bypass the signal, in this case, keeping only the information between 1 hertz and 35 hertz, so capturing from delta to low gamma, because below and after that was highly contaminated. And we had to also downsample that to 110 hertz. Unfortunately, sensors are not sampling at the same frequency, so in this case, the EEG is sampling at 220 hertz, meaning 220 samples per second, but the accelerometer is not working at that rate. So we had to align them, in this case, by resampling. And to remove artifacts, transient or high amplitude artifacts, as you see here with the jaw clenching, we use artifact subspace reconstruction, and we will it's one of the toolboxes that we had adopted in our pipeline. And then we had to remove some flat EEG segments that after the cleaning of these transient or high amplitude artifacts to get cleaner data. And then we had to segment the data into windows of 1 second with overlap to analyze it. And I'm not going to go into analysis today, but we have here a reference. I'm happy to share this presentation. So this is the typical setup for MoBI that we use in our experiments. We have been trying to be very consistent in all the experiments that we conducted in the past several years to enable both data sharing and also integration. So we typically use EEG caps with 32 channels, as you can depicted here. And of those 32 channels, we reserve 4 to track the vertical and horizontal movement of the eyes. You can see here on this participant, the vertical EOG and the horizontal EOG. So that allows to track head movement, the generators of electrical activity from that. And we are developing techniques to actually use these EOG channels not only to detect eye blinks which contaminate the signal, but also as a way to track gaze in the participants. And we hope to be able to share that with the community soon. So these are pictures with examples of the different the setup of the different EOG channels, reference and grounds. And you can see here a backpack where we keep the Wi-Fi transmitter, which takes the signal recorded in the brain and sends them wirelessly to the receptor for processing. I want to also highlight the inertial measuring unit here in the front of the head that's tracking head movement from the participant, accelerometer, magnetometer, gyroscope. So we have a 9-axis motion sensor that provides exquisite information about the kinematics of the movement in the participants. A typical data acquisition consists of recording the impedance of the electrode. So what is the quality of the signal of the electrode on the surface of the scalp? Of course, you want this impedance to be as low as possible. In the old days, they had to be under 5 kilo ohms or even 1 kilo ohm, because the instrumentation that available at that time will prevent a good recording if you have higher impedances. Nowadays, we are using active electrode where the signal is pre-amplified on the site. So rather than going passively to a wire to the amplifier, now that's happening on site, which is more robust to artifacts and this to better impedance. So we can relax now the impedance up to 50, 60 kilo ohms as opposed to 10 kilo ohms or 5 kilo ohms or 1 kilo ohm in past years. We do that at the beginning of the experiment and also at the end of the experiment, because as I mentioned before, these signals can change over time due to changes in humidity, temperature, or even the motion of the electrode and other factors. So it's very important to record at the beginning and at the end so that you can account for that during the analysis. And this is unfortunately something that is not done often. Very few studies check impedance at the beginning or the end, and some don't even report on this at all. Now, we typically use what we call a pre-calibration phase, where we have an eyes open condition, typically 1 minute, rest, and we record baseline EEG activity, and then eyes closed for another minute. And then we repeat that after the intervention. So the music-based intervention will be in the middle, right? So you perform this pre-calibration and post-calibration in relationship with the music-based intervention. Just to remind you, the experimental setup, we have one IMU sensor. Here is the black box here, which is also wireless. And we can record, sometimes we put them in the body, we put up to 16 of these sensors if we're interested in capturing also body movement. We have 28 sensors for the EEG and 4 EOG sensors. And we typically record 1,000 hertz for the EEG and EOG and 128 hertz for the IMU or the motion sensor. So this is a typical paradigm we use in our experiments. Now, I mentioned that, there are many artifacts. And an important one are the eye blinks, right? They are very strong. They are just behind your eyes, and they propagate the whole scalp. So again, here we have a participant showing the sensor that we use to track the generators of the eye blinks, eye movement, both vertically and horizontally. What you can see here on the first column, the scalp map, the top, you can see the nose here. So the top is the eyes, towards the eyes. The bottom represents towards visual cortex or the posterior parts of the head. So these plots are representing an average of the first 15 blinks. And you can see that the very strong polarity, in this case on the left, that represent these 15 eye blinks. And then at the bottom, you see the average of the last 5 blinks that were recorded in a session for this participant. Again, you see the change in polarity here towards the left. They're not exactly the same. You can see that there are different distribution, right? These are the average scalp maps for 15 trials that had blinks beginning at the end. Now, I want to bring the attention to the top right figure, which is very telling. So we have, at the bottom, we have different sensors. So we start on the left with the vertical electrooculogram electrode, the VOG, right? So that will be here on the figure at the top, right? So that's measuring the eye blink. And you can see the eye blinks, right? Very strong, high amplitude. This is 164 peak for this eye blink. And you see two measurements. One is at the beginning of the recording in light blue. So around 25 seconds from the beginning of the recording. And then at the end of the recording, about 550 seconds. And, they are more or less similar, recorded by that sensor. But as you travel on the horizontal from the top left here down to the anterior part of the brain along this diagonal, you can see how the amplitude changes. And in fact, it changes polarity going from F3 to FC1, right? And also the shape. So this distortion of the generator, right, of the eye blink recording the front is due to volume conduction, right? And that is affecting the propagation of that signal. So if you use a template-based algorithm for removing eye blinks, it's not going to work, right? Because you're going to use probably this template that you see here at the sensor electrode to remove that template from the EEG signal in the other electrodes, but they are changing. So moreover, if you compare the beginning to the end, they also change in amplitude. So it's non-stationary, it is changing. So these artifacts are not fixed. So you cannot just use a template matching algorithm and remove them. The other important challenge is that if you look on the other axis, the impedance. So this is basically a measure of the quality of the recording. Remember, the lower the impedance, the better the signal, right? So this is very good. It goes between 1 and 16 kilo ohms. For this active electrode system, if you are under 60 kilo ohms, even 100 kilo ohms, you get a good signal, right? So this is really good. You can see that from the beginning to the end, there's a change in the impedance, right? Except maybe for electrode ZC at the center. But you can see that there is a change in the impedance, certainly in the posterior part of the brain here in this area. So these three electrodes here have different impedance, and that is leading to changes in the signal, right? So if you don't measure the impedance at the beginning and at the end, there's no way for you to know whether the signal is not contaminated by these changes, right? Might be due to, again, to temperature, the gel drying up, the humidity, and other factors. Now, at the bottom of this plot, you see the raw contaminated EEG. Now compare that to the activity due to the blinks alone, right? So these eye blinks are on top of the brain signals here on panel C, right? Now, once you are able to detect these artifacts through our algorithm, which is shown here on the bottom left, then you can identify the artifact amplitude distribution of all the eye blinks, right? And then be able to remove them in an electrode by electrode basis and also sample by sample basis, so that you get artifact-free EEG. And of course, this signal is very tiny here, because we kept the scale from -100 to +100 microvolts, but the EEG signals are in the order of 10 microvolts. So it's really, the eye blinks are 10 times as larger as the brain signal you want to record. Now let me go back to the algorithm that we have developed, which is an adaptive noise canceller, right? So identifies the artifact at each electrode, at each sample. So it's a sample adaptive, which means you can use it both online and also offline applications. And I'm going to refer you to the paper, which is published in Journal of Neuroengineering. We compare this with some of the most widely used methods for removing artifacts, including independent component analysis, which is a blind source separation algorithm. And we outperform those algorithms. So this is something that we use in every single recording. So basically, you have at the top, you have the contaminated EEG. That contains, of course, the EEG, the artifacts due to the EOG, any bias in the signal, changes, drift in the potential due to many factors, right? So all of these is included on the EEG, right? Because as these performers were using this, as the individual were using this, we didn't include, and the task was constrained, we didn't include all type of artifact, but this is basically, eye blinks, biases, and drift in the potential. Now, we developed an adaptive filter that, as reference, is given the vertical and the horizontal eye blink, and also a constant. And that constant is used to learn these biases and drift that are very slow over time, or sudden changes in the step on the EEG, right? So basically, tracking those transient or gradual changes in the EEG that are due to other factors. And so the adaptive filter has the function of using this reference to learn the artifacts. And when it has learned the artifacts, it removes the artifacts from the contaminated EEG so that you have clean EEG, right? So in that way, we are able to clean this. And remember, this adaptive filter is operating at each time sample and at each channel, each sensor electrode. Now, the other important artifact is due to motion. And we conducted an experiment, also published in Journal of Neuroengineering, where we had participants wearing an EEG cap, as you can see here at the top left. Of course, had also the vertical and the horizontal EEG sensors. It had the 9-axis IMU. But on top of that, we use motion tracking. You can see the camera, the optical system, to track individual movement of the electrodes as the participant was walking on a treadmill. So we were able to track the overall head motion through the IMU and the individual motion of 4 of these sensors, trying to understand what was the relation between the global effect of motion and the specific effect of that motion on a single electrode. Now, what we did was to augment, to expand, to generalize the method that I showed you before, which we call the H infinity, to account for motion artifacts. Now, if we go to the right, we have representative data from a participant walking on the treadmill at 2 miles per hour and 4 miles per hour. So a little bit faster, in fact, twice as fast, right? And you have at the bottom the different phases in the gait cycle. You know, I was stepping on the treadmill, and then we had a frequency. You know, in this case between 0 and 18 hertz. We see that at 2 miles per second, right? There's a bit of an artifact in the lower frequencies, right? In the delta band. But as the participants started to walk twice as fast, we see a more prominent artifact in the EEG signals in this plot that were due to the walking, to the motion. You can see that better at the bottom plot. So we have 3 signals here. We have rest, so no motion. We see the 1/f curve, and also the typical peak in the alpha range around between 8 and 12 hertz. That is commonly seen during EEG capture at rest. And then we have the EEG collected during walking, and you can see the prominent peaks here introduced due to the walking. Right now, if you go to twice as fast, then it becomes really clear. You see the fundamental frequency of walking that is causing an artifact on the motion, on the EEG signal, right? But also you see harmonics, right, that contaminate the signal. And so by using our augmented adaptive noise canceller, we are able to identify and remove these peaks from the signal so that we have a clean signal that is not affected by the motion artifacts. Now, going back to the model, what we did was to add a Volterra kernel, basically a polynomial of second order, to account for these harmonics, right? So the H infinity filter that we first developed in 2016 is a linear system. But to be able to account for the motion artifact, we had to add a nonlinearity here. So the motion artifact filter is a nonlinear system. That means we need to take care when we apply this system, right? And we have developed a pipeline that will come in a few minutes. So this is just to it's good news. It means we can collect data even during motion, right? We need to be, we need to be careful, and we need to look at the signals, but we have a way to identify and remove them in a sample by sample basis. If we want to do this online, say during a performance in a active music condition where you are playing, or when you're listening to it. So this is a processing pipeline that we have developed in my laboratory. And in addition to EEG, EOG, and motion, we also collect video, 24 frames per second or higher if available. And all of these are synchronized. And I will talk about that a bit later. But when you are running a MoBI study for the first time, right? It's always good to inspect your data, right? And this is something that is really important. You can overemphasize, right? Because these are algorithms that that have been developed and they have been validated, but we always have new type of protocol, new conditions. So it's good to inspect visually through the video, and also using the motion sensor. So we typically label data during the recording, during the session. We can insert triggers as well. We can insert labels in real time as we are recording. We perform a technical review by visualizing the video, the EEG, the EOG, the motion together, so that we can identify potential sources of artifacts, and then we can compare with the output of our algorithms. And because the sensors are operating at different sampling frequencies, we typically need to resample, right? So you can bring the 128 hertz, you can upsample that to be in the same rate than the EOG and the EEG, for example. Now, once we do the visual inspection and review, we enter the first stage of artifact removal. In this case, we use the Edge Infinity. And the Edge Infinity filter that I mentioned before has a triplet of parameters, and these parameters need to be provided. And on the paper, we provide some values. And as we have performed this in different type of tasks, as you saw before, during writing, during dancing, during appreciation of art, during music production, we have developed information about what's the best set of parameters that you can use to maximize the cleaning of the signal. But it is something that also needs to be checked. And then we have robust re-referencing that can be used to ensure that the average reference signal is consistent, right? Which is affected by artifacts, of course. And so we use that in a pipeline. We use a bandpass filtering before the signal is sent to the motion artifact filter. So basically, we are removing very low frequency and very high frequencies that are outside our scope of analysis, or that we know that are highly contaminated. This depends on, again, depends on the task that you are conducting the protocol. Then we go to the stage 2 of removal, where we use artifact subspace reconstruction. Again, this is typically used to identify and remove transient or high amplitude artifacts that were not captured by the other filters here, and has one parameter. 15 is a typical value. You can find the literature range. We recently have developed a deep learning approach to automating the selection of this parameter, which is very important for the operation of this ASR model. Once we have gone through this pipeline in continuous time, right, we are ready now to look at the data from our video, not things that we know that are badly affected, or and then compare with the output of our cleaning procedure and identify any noisy data portions that were not cleaned. But we have been able to increase dramatically the percentage of data you can retain due to the filtering. And sometimes you might need to reject some segments that were not clean or that are really affected. Sometimes, it could be that for some reason the battery for the Wi-Fi transmitter got depleted, and then you have no data, right? And so you might need to then remove that data, right? And so then the final stage of artifact removal, we use independent component analysis. And this is really to identify residual artifacts that we don't know the precedence, that we don't have reference signal for. And we integrate that with dipole fitting using either the MRI, the magnetic resonance imaging, the structural MRI from the participant, if available, which will provide the more accurate source analysis, which entails given a collection of measurements of EEG on the scalp, what is the most likely source for inside the brain that led to that EEG, right? And in the absence of MRI for the participant, then we use the MNI template, the Montreal Neurological Institute MRI template, which is used widely in the scientific community. And then we use clustering algorithms to aggregate these dipoles and localize them in Broadman space, so that we can label these sources with a particular Broadman area, so that we understand the network that has been recruited in a particular protocol. And sometimes you find dipoles that are artifactual, right? That some noise component that was not identified by our procedure. Then you have here an opportunity to analyze that and remove it. And the output of this pipeline is the clean EEG, right? So this is a process that has been automated to some extent. Again, there are some parameters that you need to verify, and the dipole analysis is an important part of this. So the final challenge, well, not final, but one important challenge is synchronizing all the sensors and the equipment. And this is particularly important when you want to conduct experiments with dyads or in a group setting, right? Think about a participant and the music therapist, where both of them will be wearing the cap, or maybe in a social context when you have two participants that are being recorded simultaneously, they are interacting during the therapy. And in general, any group setting where you have more than two people, right, where you want to understand how one person influences the other during the intervention, right? And there are two ways to do this. One is by hardware, and that's the way that we prefer. It's very accurate. It's an electronic pulse that's sent to all the hardware to synchronize the recording with very high temporal resolution. Or you can use software synchronization, right? There are tools available like Lab Streaming Layer you can use to synchronize different systems. And of course, you can do a post hoc synchronization by manually marking corresponding events in each data set. That's time consuming, is really not something that I would recommend doing. So you have hardware synchronization that's preferred. Next thing will be a software synchronization. But again, it's software, so you need to inspect that. And finally, manual. So just to give you an example, this was the setup that we use a protocol where we were recording from 5 participants simultaneously. This is called hyperscanning, where you're scanning multiple breaths at the same time in synchrony, right? For this experiment, we had 4 participants that use this cap, and we have one that used a different system. In this case was the Nautilus G-Tech that included a accelerometer on the cap. So these are from different vendors, and we had to combine them to be able to record 5. For the brain cap, we have a transmitter, and this is in Wi-Fi that basically is sending this signal through these antennas to the receivers. And then we have, of course, also the transmitter for the other cap. We have 5 IMUs, so each participant had a motion tracker for the head, running 120 hertz. So we have 5 caps, 32 channels. 4 of those were used for tracking eye movements, and then we have 5 IMUs to track their head movement. And then we have a trigger, a minor trigger. That trigger went to all the devices to make sure that we were synchronized and that we could analyze, brain-to-brain synchronization among all the performance. The trigger is also used to synchronize video. We normally do it optically so that the camera can see the LED flashing 3 times, different times during the therapy or the performance. So we synchronize the video, the EEG, the EOG, the accelerometers, and in some cases, the output of the program that is handling this through a brain-computer interface can be used to control machines, for example, real-time projections. So I'm going to give you an example of an experiment that we conducted a few years ago. In this case, we have 3 musicians playing jazz, a drummer and 2 sax players. We conducted 3 trials of this study. Again, you remember we have the pre-calibration, impedance, eyes open, eyes closed at the beginning of the experiment, at the end of the experiment, so that we can look at changes in impedance and other things. And then we have a protocol where each of the musicians will start the trial and play alone for 5 seconds. So in this case, trial 1 was P3, trial 2, P1, trial 3, P2. So this was counterbalance, right? So we have P3, P1, P2, P1, P2, P3, and P2, P3, and P1. So we have all the possible combinations, right? On the right, we see the impedance at the start of the experiment and at the end of the experiment. So, this is a really good recording. You can see there are only a few electrodes that had impedance greater than 60 kilo ohms, which is our threshold for for accepting good an electrode. There were a few that were about 25 kilo ohms, but less than 60 kilo ohms, so it's still usable. And in this case, P2, we had, by the end of the experiment, everything was working really fine. All the electrode impedance were great. So this is good. So in this experiment, which lasted about 45 minutes, we had really good signal quality. And you can see here examples of that. This is a raster plot showing the magnitude is 800 microvolts. This is the scale of the eye blinks. You can see here the eye blinks in the horizontal and vertical sensors. Remember, these EOG channels are used as a reference for the adaptive noise canceller. So it learns how this signal is affecting each portion of the scalp and is able to remove those in a sample-by-sample basis. Here we have 5 channels of EEG, right? We can see here some artifacts on OZ, which is at the bottom of the skull, in the central part. We see some noise here. And of course, we have a recording of the sound because they were playing, right? We want to be able to analyze the sound and analyze the brain activity with respect to the sound and the movement. So this is an example of an experiment, in this case, active music production and interaction. So these are some examples of what they did. So that was a solo that he was improvising, playing the sax. Now we have a duet. So we have now two people that are interacting. Remember, all of them, all the three are being recorded in synchrony. They're synchronized. So this is the duet. So the music was rated by experts in music composition from the music college, and so they rated them as synchronized or unsynchronized. So this was an example of synchronized recording. And the last one is when all of them were playing together and they were not synchronized. So that's an example of each of the segments and synchronized and synchronized. Right. So what we did, after cleaning the signals for different type of artifact, we decided to quantify the level of interbrain neural synchrony using the bispectrum. That's so when looking at this, of course, there are different ways to look at quantifying synchrony. Most basic, just a simple correlation, but these methods have their benefits and advantages, and we think that the bispectrum estimation methods is some of the best methods out there. The higher bispectrum magnitudes at a given frequency pair reflect non-random interactions, phase coupling, and nonlinear multi-frequency interactions between the channels, right? So this is the equation, and this was computed with respect to the eyes open condition, right? So this is our baseline, and so we computed the level of neural synchrony between the pair, the dyad, at different frequencies, different time segments, and pairs. So let me tell you a bit about the outcome of this. So here we have on the right, so this is bispectrum, it's a frequency by time plot. At the bottom, we have minutes. Each segment lasted for 15 minutes. We had 3 segments. Each lasted for 15 minutes and consists of 3 segments of 5 minutes, right? So here we have P3 starts. So this drummer, this sax player started playing, and we are computing the interbrain synchrony between P1 and P2. They are not playing, they are listening, and they may be paying attention to the to the performer, right? And you can see here the level of bispectrum, right? The level of interbrain synchrony along the frequency range from 0 to 50 hertz, right? So going to the gamma range, from the delta to the gamma, right? And we see that when they are not playing the linear synchrony, there's some at times, but as soon as the P2 joins P1, right? We see an increase in the level of synchrony in the gamma range, right? So that means that as they are both playing together in synchrony, you see an increase in the interbrain synchrony between those players. If we repeat that for the other diet 1 and 3, in this case, P3 starts, so he's playing alone, no synchrony, but then P1 joins, right? And then we see the increase in synchrony, and it remains there for the end because they kept playing together, right? And then the last dia was P2 and P3. Again, that only happens at the end. So again, you see level of synchrony just at the end. Now, you notice you compare trial P2, P3 at the bottom with P1 and P2, that there's a lot more synchrony at the top in the gamma range, more consistent, higher level of synchrony between them, right? We learned after the fact that P1 and P2, they used to play quite a lot together while P3 was playing with them for the first time, reflecting less level of interbrain synchrony between the unknown player and the others, right? Which was very interesting to learn. Now, this represents the topographical representations between participants' intersynchronization in different regions of interest, which are marked by the labels here, from the occipital to middle frontal, central to anterior frontal areas. And what you see here when you compute, when you assess the number of intersynchronized ROIs based on the bispectrum for both the gamma and the beta range. So these are higher frequencies, lower frequencies. And you compare that as a function of passive listening, passive versus active, or both members of the dia playing. We see that a network starts to develop, and mainly in the posterior part of the brain, in the sensory areas of the brain, right? So remember, they're improvising, and so they are using vision and audition, right, to guide their movement. Of course, there are other factors, the mood, the memory, and everything that goes there. But it's very interesting that if we look at interbrain synchrony between dyads, right, we see that when they are passively listening or observing the other, there is some level of interbrain synchrony, but is very weak, and it's primarily in the posterior part, is sensory driven. As one of them becomes active, you see this network growing, and in both the complexity, the different areas of the scalp, and also on the strength of that synchrony, which is dominant in the gamma range. And you start to see also some anterior projections, some decision making that probably one of the performers is making, right, as he decides what to play or what to do. But then when you have the both active, the network becomes very clear, and you can see both the posterior network and the anterior network. So this is telling us what are these networks, right, that were recruited during this improvisation. And from this, you can do source analysis, for example, to understand where this signal is coming from. So you can repeat this using the dipole space in terms of the Broadband areas, and many other. And you can also ask questions about whether you can predict the performance of the other based on the brain activity of the observer or the listener or the performer. So to conclude, I believe that EEG-based brain-computer interfaces are powerful tools to understand the brain in action and in context in real-world settings, and they can be applied to the delivery and analysis of music-based interventions. Second, quantification of coupled brain behavior can inform movement-based intervention. social and affective neuroscience and other areas. So this is a great opportunity for us. BCIs also allow the study of music as a neuromodulator to inform music-based interventions. And we will show next, a hands-on setup of this technology. And we will, at a different time, we will focus on further analysis and how we can use that analysis to ask questions about mechanisms. So at this point, I'd like to stop this presentation and answer questions, and then we move to the lab. So let me see if there are any questions. Or maybe, Julian, you can help me to okay, so there was answer, right? Some questions were answered during the lecture. Great. Before we move to the lab part, and my students are ready there to conduct that hands-on. I want to mention that it is really important to have the right team. It's the team science, right? Because as you can see, the technical aspects can be demanding. It's very important to develop experience in the setup of the system, because as you have more experience with the setup of these caps, for example, your impedance recordings, your impedance measurements are going to get better, reflecting that expertise. So of course, there are published best practices by societies, and also we have a publication on best practices as well, and we would share that with the community. So if there are no questions, we're gonna switch to the lab, and so I'm gonna turn off my camera and go to the lab, and then we can have an example down there. Julian, I don't know, you want to stop the recording now and then start a new one?
We'll just keep recording and do the editing afterwards.
Okay, great.
Okay, so we will start with the second part while we wait for Dr. Contreras. I think you all know me. I am Anel, a PhD student here in ECE. And well, as Dr. Contreras showed in the slide, the setup is divided into different stages. We have from the stage of EEG, where we have our receivers in the desk, we have our transmitters with our subject, and we also have the stage of the opals, which capture the motion or the head of the persons we're recording, but they can also be used to capture, for example, arm movement, leg movement, and magnetometer and gyroscope. So we use these signals and then we synchronize them with a trigger box, which is hardware that was designed here in the lab. This hardware blinks light, and we will talk a little bit more further in the workshop process. So
well, hello, my name is Joshua Lima. I'm also a second year PhD student here in the lab. I work under Dr. Contreras. So in my case, I'm just gonna talk a little bit about how we actually start implementing the setup in one of our participants, normally during an experiment. Well, basically what we have is that we first need to measure the head. So this is just in order to follow a standard that is called the 1020 International Standard, that is for the location of the electrodes. So this consists basically on measuring the head of the participant from the nation to the inion. where we actually do this measurement, and then we need to select the 10% of this measurement in order to start locating all the electrodes. But also before doing that, we also need the circumference of the head of each of the participants, because the caps that you normally see here, so they come in different sizes based on the participants. So we have from normally pair numbers like 52, 54, 56, and all the way. So depending on the head of the participant. So, okay, I'm gonna, here's one of our students. So he volunteered to show the measurements. So first what we do is like we place, as I mentioned in the nation, the first part, the 0, and then we go all the way into this bone that we have in the back part of the foreheads. So in this case, it will be 34, and we need to put a little mark on the 10% of 34, so it will be 3.4. So we use normally a marker. So this is just to put a little mark where we are gonna place the electrode. So yeah, once we place the mark, we need to actually start putting the actual setup with the EEG cap. So in this case, our participant has a circumference of 60 centimeters. So that's the cap that we're gonna show, or we're gonna use for with the participant.
So in this case, we are using the 28 electrodes to capture the brain activity, the 4 for the eyes, and you can vary the positions where you put them. Right now they are in the 1020 system, but for example, if your project focuses on recording only from the motor activity, you can put in the central lobe of the head. You can also focus in the occipital part, but in this case, we are doing a more general setup. So we're using all those electrodes. These caps also come in different numbers of electrodes. So you can also have caps with 64, 128 electrodes, and it all depends on your focus and your goal.
Yeah. Also, it's important to mention, like, for example, here in this part, like at the top, we also have something that is in order to place the inertial measuring units, the opals, that we use in order to measure the head acceleration. So once we have this, we start placing the cap based on the mark where we put it.
Can you strap your
So it's a medium cap, medium size.
It says 60 centimeters. Yeah.
And then the electrodes that we use for the eye activity are the electrodes that are more outside in the cap, so we can also use them this way. And what we do is that we slide it through the cap just to make them secure, and they don't fly out when they are doing these types of exercise. For example, we record from dancers, from musicians, which are very active, and sometimes they forget they have the cap because they get used to all this equipment, and this allows us to record creatively what they are doing and just in their normal routine while they perform the arts.
Yes. So, well, in this case, once we have fitted our participant, the next step will be actually to start measuring the impedance. So, as and Dr. Morales was mentioning during the presentation, the impedance is the position of the signal to for the EEG to go to the scalp. So in this case, what we use is a control box that we connect all the electrodes together with the ground of reference, and it will display some colors. So these colors will depend on the level of the impedance that we want to achieve. So normally, when we have a red color, it means that we have a bad impedance that it will be about 60 kilo ohms. And then what we want to actually achieve is normally below 20 kilo ohms.
We place the ground electrode in the left ear lobe, and the reference in the right earlobe. And well, that's our main setup in this case. And you can see the colors of the lights. They start with red, and then they start changing as we apply this gel, which is a hypoallergenic gel. It's high viscosity electrolyte gel for active electrodes. So these are active electrodes. And what we do is that we use these syringes and we start moving a little bit the hair inside each of these electrodes. And then once we have like a good contact, we put a little bit of this gel. And we also do that for the ones in the eyes. We have these stickers too. So these stickers allow us to put the sensor in its place for the ones in the ears. We just put it like this. And once it is like this, we also slide it through the cap. So something very important while you're doing this setup is that the participant has to be in its chair the whole time. And you have to also tell them some specifications. For example, the day of the project when you're gelling him or her, she has to have the hair clean and dry. She also has to know wear makeup that day, or she could wear makeup, but then when you are applying the cap, you will need to use these alcohol pads to remove the excess of oil of the skin. And this allows us to also contribute in having a better impedance and signal quality while we are recording. So normally what we do as first step is that we clean with these alcohol pads. We let it dry. And we apply just a little bit of gel, because if not, you can also create a bad conclusion. So it's only a good amount. You begin first with applying the ground and reference to have this initial step. And once these ones are in good signal, so for example, in yellow or in green, that means that you can start gelling all your other electrodes. The caps also come in different colors. So sometimes the artists prefer to use white or black, and you can customize that, as well as the colors of the holders.
So in this case, once we have placed like the electrodes for ground and reference, what we do is that since we have a lot of movement normally during artistic performance, or we actually want them to be placed, we normally use a tape. So this tape allow us like to keep the electrodes in the right position, so they actually we're sure that they are not moving.
As for the one of the eyes, we select the channels as up, down, and then left temple, right temple. But when we apply it to the subject, what we do is that we ask the subject if you can look straight to the front. And then when she's looking straight to the front, we find where the pupil is at. And from there, we try to align the electrode with the level of the pupil so we can get a good signal of the eye blink. So we can later use in our cleaning filters in a pre-processing step. Also, before applying this, we also use the alcohol pad just to make sure the area is clean and dry.
I don't know if it's possible that you can hear that noise. So normally during the experiments, when we are actually measuring the impedance, the control box has like a saving energy, so it just turn off all the electrodes. So we need to restart in that case the system. And then we will be able again to see all the colors for the electrodes.
Yeah. Well, somebody in this room would like to try the gelling part? Yeah, you can.
You're welcome to.
Oh.
So in this case, what we normally do when gelling is that we place the syringe slightly, and then we start placing just a little amount. And then we need to wait some time. We do, like normally, in order to electrode to have a good contact, you need to move a little bit the hair with the needle. Just being careful that not to damage the skin of the participant. And for example, for the ones that are close to the eyes, like part of good practices will be like placing the hand like this, and then just putting the gel like again, and then yes, like you will put inside the electrode, like there is a space between the electrode and the like this whole thing. I'll show you maybe.
So you have to put like a line of this for each electrode. And you have to be sure you're moving the hair before you apply the gel. So with the tip, you have to do like to the left and the right. Very superficial, like I'm not pointing to the scalp. You have to do these like circular moves around. And apply the gel. During our different performances, sometimes we have to adapt to the venues. We have to adapt to the, for example, concert gowns or clothes they are wearing. So we use fanny packs to hold the rest of the equipment. That is the transmitter that the subject will be wearing with the cap. For example, sometimes they put them here in their bags. They can also wear them like this and use the electrodes here. So they can work with our technology and also feel comfortable with what they are doing. Yeah.
Okay.
Yeah. Another good practice for this is that also before you gel the subjects, you have to ask if they had coffee during the day, because we normally restrict the consumption of coffee before recording the signal, since it can alter the result that we're getting. And this control box that we have here works with lights that are powered by batteries, but also once we finish to gel all the electrodes, we can also power with the computer. So the computer has the power to open an app and then watch what are the actual values for each one of these electrodes that we have. Does anyone have any doubt up to now? Yeah.
No, I just didn't hear what you said.
Oh, that if you have any doubt of this setup of the equipment.
I actually have a question. So you said after you measure from the eyebrows to this bone,
Mhm.
do you also measure the circumference to see what size cap?
Yeah, so at the beginning, we measure the circumference to set the size of the cap. And once the size is set, then we set the placement of the cap. So every time we put the cap on this person, it will be the same measurement, and we know we are recording from the same points.
Okay.
And you, okay, but I'm gonna show you this and then I'll do it once. So, yeah.
Just told me to, like, double speaking.
So this is like the demonstration of how it is supposed to look like. But beforehand, we've set up Aditi.
What's that?
That's
Aditi.
Oh, well, we have set up her.
Maybe she can come this way.
Yeah,
and
you
now
can
let
come
me
this
just-
way.
Okay, so normally, what we do after she's doing the setup, we disconnect her. Well, first we turn her off. Then we disconnect everything. And we normally, what we do is that we use a funny pack in order to store all the cables. So,
If you can stand up.
yeah, you can stand up, no problem. You're pretty good.
Then we will need your arm to slide through the part. And then you can strap like this. Once all the electrodes are green, we do this. And they are supposed to look like this. So what we're gonna do now is that we're going to connect her to the control box. So you can see the levels of impedance of her cap.
Now you're good to make it.
Before and after the presentations, we go to their devices in this part. Or maybe we can go to the front to the camera. So from here, we take out the ground, the reference, and the bundle, which has the electrodes. Then we connect it to these plugs. And we use this cable where the batteries were. To use the second option of the control box where allow us to see the results on the screen. Can you hold these books?
So once she is connected, what we do is that we use a software called ActiveCap Control that basically we use in order to measure or store all the impedance values. So in this case, you will see like this is normally the screen for the where we can see all the channel locations. And then we go to impedance check. And we need to actually click on impedance on so we can actually visualize all the colors for the values of the impedance. So in this case, like since he has been for a little bit of time with the cap, we have some of them that we need to be fixed. So you will see basically the same definition as I was mentioning before, like we could have different colors between green to red, and the red ones are the ones that normally are with a bad impedance.
Yeah.
So in this case, in order to fix them before the experiment and we identify all of them, and then we click on save impedance and we identify the name of the channel. So in this case will be F3. And then in this list, we identify the channel that is F3. So it will be number 4.
Mhm.
And then we will fix it in the
And as you can see here in the cap, number 4 is red as well as number 8. So these two that are here correspond to the two you were watching in the software. So we go with the syringe and we reapply the gel, and we see how it was red and now it's green. We do the same with the other one. And now you can see in the screen how all are between green and yellow, which is below the threshold. So when they are like that, we go and save them.
we click on save, we do exactly the same procedure, check which are they. And then what we normally implement is like we copy all of the values and then we create in a notepad. And then we paste all of them and then we just store them. Based on the experiment, we normally create like, for example, in this case will be workshop. And then we always add like a label for pre. So in order to identify it is pre-experiment because we store these values for pre and post experiment. So we can check then the changes across time.
And what this helps us for is just to check out the signals, and also it allows us to do some graphs showing what were our signals before and after. And that way, when we're analyzing our signals, for example, in EEG Lab or other types of softwares, we can see if any channel needs to be interpolated before we do the analysis or any additional metric we need to put into our data set for it to be clean.
So once we have checked the impedance, the next step will actually to start connecting the EEG cap to the transmitter. So this will be the transmitter that will help us to send the data to this receiver. So once we send it to the receiver, we can actually visualize the data on the computer. So we'll see that in a moment.
We connect it to the adapter, and then the adapter talks to the transmitter that at its way talks to the receiver via Wi-Fi. That's why we also have the antennas here. We turn everything on, and we will do a quick demonstration of how the signal looks like. We know they are communicating because we can see the green light here and there. But if we turn this off, you can see how it turned into blue. And that means that the connection is not that good. It can turn blue for many reasons, but one of them is that the transmitter is off. This light also turns red, but the red means that it is on, but it is not getting the proper signal. So then we apply this to the panic pack. Do you feel comfortable with like any cable pulling or everything?
Normally, we ask our participants just to verify that the equipment is well set up for them, like they are comfortable with it. So we can actually prevent any damage of the equipment and also to any problem for the artists if they get injured or something. So once we have everything set up, we are ready to look for their signals. So what I'm gonna show you here, we use the brain vision recorder application, so we will see the actual brain signal that like we are acquiring. So in this case, well, I'm just gonna ask Disney, she can like blink during 5 times, and we will see how these little jumps represent like the actual eye blinks while Disney is doing the activity of the eye blinks. Also, if you can please clench your teeth, please.
I can one.
Please.
Oh.
Doing it. So we will see also the muscle activation while she's clenching her teeth. So
So these are signals that are noise, but what we do is that the electrodes are capturing this noise and then we use it to remove. For example, with the H infinity filter, we use the blinks that come from electrode 20, from 15, 31, and 32. That if you can see those lines, can you blink again? So if you can see those lines, those are the electrodes that have like the more defined peaks in the screen. Those are the inputs for the filter noise. And also she can move and she can do any type of movement. For example, we use these in
stand?
dancers. You can stand up and walk. And the interesting thing of this is that in this lab we have 3 of these systems. So we can equip three participants and use hyperscanning to record from them at the same time, synchronize the signals, and also analyze them. So we can study the brain during these social interactions. Oh, so there's just one last item missing, and this is the opals. The opals are the inertial measurement units that you can see here. And to compute them and configure them, we use the software that is called Motion Studio. We have, every time we record, we have to apply a new configuration. And what we have to do is only dock the sensors in this docking station that is here, which is connected to the computer, and it is also connected to the antenna. Then the antenna is also connected to the computer. So once we have this, we just scan the software. We make sure that everything is connected. What we do is normally we use a multi-port. We use a multi-port and connect the antenna. We connect to the trigger box and to the opals. Once they are here, we also make sure that the opals are connected to the light to power this docking station. And then we scan the software with the application. So right now it's scanning the software. And here we have one access point, 3 docking stations, and the 3 sensors. We normally use the wireless channel 40, and this system is sampling, has a sampling rate of 128 Hertz. We click on configure and it starts configure the 3 sensors that we have here to that configuration.
that's what we should in the chat actually. We have what happens when the subject wearing the electrode cap will sing. Oh, when they will sing. Okay, perfect. So, well, in this case, well, we will record a signal, and actually the idea will be like actually placing some electrodes around the clinic, so we can actually record that part. So we can implement a filter to clean that. But normally during the experiments, we ask the participants not to actually see it. So that will be like the idea for now.
And well, while we sync these devices, we have to wait for them to blink at the same time. So you can see this green light here that is blinking. Not that visible in the video, but they are blinking at the same time. So you can see here, these are the graphs that show the signal. So you can see that when we move the sensors, the signals are also moving. So as the final step in the caps is that we place those ones in the forehead. And that is all our setup.
So in this case, it is always important to put them with the light going up and the left. So we can actually have all the same standardized for all the experiments.
Yeah, and this is our final setup. If you have any doubts of any step, you can tell us. And yeah, normally we record from 2 people, so this is how it looks like, the 2 subjects performing or playing music side by side
Or interacting.
or interacting.
But thank you for volunteering.
Yeah.
Thank you for all the viewers. And you have any questions, just feel free to reach out.
Okay, well, thank you. Is there any question in the chat?
No, I think that was the only one.
Okay. Well, thank you for attending the workshop. Uh,
Yeah.
Okay.
Perfect. So thank you so much for attending and be in touch.